Dear students, welcome to all of you. Today we are here to discuss the topic formation and structure of secondary xylem. In vascular plants, xylem is one of the two types of transport tissues, phloem being the other. The word xylem is derived from classical Greek xylon, means wood, and indeed the best known xylem tissue is wood. Though it is found throughout the plant, its basic function is to transport water, but it also transports some nutrients through the plant. The internal transport system of vascular plants, that is tracheophytes, comprises specialized tissues made of elongated tubular cells termed xylem and phloem. Xylem transports water and solutes from roots to leaves, water being essential for photosynthesis and maintaining turgor pressure. Phloem transports complex organic compounds, notably sugars, that are synthesized by photosynthesis and its subsequent reactions. As vascular plants colonized the land in the early Paleozoic and subsequently diversified in the Devonian and Carboniferous era, many of them became much larger in stature and height. For plants to grow taller, their transport systems had to develop improved structural support and, in the case of xylem, higher conduction efficiency, so that water could rapidly reach all the way from roots to the topmost leaves. Better support for this was achieved by rearrangement of specialized supported tissues, for example, parenchyma, sclerids and fibers, rearrangement of vascular tissues into vascular bundles, and deposition of lignin as a rigid secondary cell wall material in metaxylem and secondary xylem. Secondary xylem forms the woody tissue of shrubs and trees. Primary xylem is the xylem that is formed during primary growth from procambium. It includes protoxylem and metaxylem. Metaxylem develops after the protoxylem but before the secondary xylem. It is distinguished by wider vessels and tracheids. Secondary xylem is the xylem that is formed during secondary growth from vascular cambium. Although secondary xylem is also found in members of the gymnosperm groups, Gnetophyta and Ginkgophyta, and to a lesser extent in members of the Cycadophyta. The normal process of growth that occurs in every plant body is known as primary growth. It is the result of activity of primary meristem. The process of primary growth results in the formation of primary permanent tissues such as primary xylem, primary phloem and primary cortex. However, in dicot plants, there is a process of growth that begins after a known period of primary growth. Such a growth is known as secondary growth. It is the result of the activity of secondary meristem. It results in the formation of secondary permanent tissues such as secondary xylem, secondary phloem and secondary cortex. As a result, secondary growth brings about an increase in the girth of plant body. In a dicot system, secondary growth occurs both in the stele and cortex. The process occurs simultaneously but is caused by separate strips of secondary meristem. In the stele, secondary growth is initiated by vascular cambium, while in the cortex it is initiated by core cambium. Now, first of all, let us take up secondary growth in the stelar region. It is the result of activity of the vascular cambium which occurs in between xylem and phloem of each vascular bundle. Hence, it is also known as intravascular cambium. In addition, towards the beginning of secondary growth, there is a process of redifferentiation in some of the parenchyma cells of the medullary rays adjoining the vascular cambium. As a result, these cells now become meristematic and represent the interfascicular cambium. The meristematic cells in the intravascular cambium and intervascular cambium fuse and result in the formation of a continuous strip of meristem called cambial ring. The cambial ring at this stage has primary xylem on its inner surface and primary phloem on its outer surface. The cambial ring exhibits mitotic activity on both the sides. The mitotic activity on the inner surface results in the formation of cells which differentiate into a set of xylem. It represents the secondary xylem. Similarly, the mitotic activity on the outer surface results in the formation of cells which differentiate into a set of phloem. It represents the secondary phloem. Due to the formation of secondary xylem, the primary xylem becomes pushed more towards the pith and pith gets slightly reduced. However, the secondary phloem grows and completely masks the primary phloem, hence it is not visible. The mitotic activity of the cambial ring is purely seasonal. It occurs only twice during every year, once in the spring and once in autumn. Thus, every year 2 sets of secondary xylem and 2 sets of secondary phloem are formed. Each year the mitotic division of the cambial ring usually begins in the spring season. The secondary xylem that is formed in the spring season is therefore known as spring wood or early wood, while the secondary xylem formed in the autumn is known as autumn wood or late wood. The spring wood is generally characterized by the presence of xylem vessels having wider lumen. This is because spring is the ideal season for the growth and water requirement of the plant is more in the spring. The autumn wood has xylem vessels with narrow lumen since water requirement in the winter is less. The 2 distinct layers of secondary xylem, the inner spring wood and the outer autumn wood, together represent an annual ring. One such annual ring is added every year due to secondary growth. Thus, it is possible to ascertain the age of a dicot tree by counting the number of annual rings. While every year 2 sets of secondary xylem and 2 sets of secondary phloem are formed, only one set is visible because the secondary phloem formed later, that is in the autumn, grows over and masks the secondary phloem formed earlier, that is in the spring. This is however applicable only to the temperate regions. The growth seasons in the tropics are different, with the growing season coinciding with the rains. In true rainforest areas where seasonality is least pronounced, many trees show continuous growth and growth rings are not seen at all. Thus, secondary growth in the stele results in the formation of secondary xylem and secondary phloem. Due to the addition of these tissues, there is an increase in the girth of the stele. As secondary growth continues to occur, several changes take place in the secondary xylem. In a much older stem, the secondary xylem or wood shows 2 regions, namely duramen and alburnum. Duramen, also known as heartwood, represents the centrally located inactive portion of the secondary xylem. It appears dark in color due to the accumulation of pigments resulting from oxidation of organic compounds like oils and tannins. It is incapable of conducting water and provides only mechanical support to the trees. It is this part of the secondary xylem that is used as the commercial wood. Sapwood, also known as alburnum, refers to outer living layers of the secondary wood of trees which engage in transport of water and minerals to the crown of the tree. The cells therefore contain more water and lack the deposits of darkly staining chemical substances commonly found in heartwood. Sapwood is thus paler and softer than heartwood and can usually be distinguished in cross sections as in tree stumps, although the proportions and distinctness of the two types are variable in different species. In secondary xylem, both axial and ray parenchyma cells located next to vessels may form outgrowths through pit cavities and into lumen of vessels when the latter becomes inactive and lose their internal pressure. These outgrowths are called tyloses, and the parenchyma cells that give rise to them are referred to as contact cells because they literally are in contact with vessels. Tyloses are often induced to form prematurely by plant pathogens and may serve as a defense mechanism by inhibiting the spread of pathogen throughout the plant via xylem. Alburnum, also known as sapwood, represents the peripheral active portion of the secondary xylem. This portion is still capable of conducting water. Due to its meager mechanical strength and lack of durability, the sapwood cannot serve as commercial wood. Wood is divided according to its botanical origin into two kinds: softwoods from coniferous trees and hardwoods from broad-leaved trees. Softwoods are lighter and generally simple in structure, whereas hardwoods are harder and more complex. Hardwoods may be divided into ring porous and diffuse porous trees. In ring porous trees, the vessels laid down at the beginning of the growing season are much larger than subsequent vessels laid down at the end of the season or ring. Diffuse porous trees form vessels of roughly the same radial diameter throughout the growing season. Larger vessel size permits more rapid water conduction. Now, secondary growth in cortex. It is the result of the activity of a secondary meristem called core cambium, which appears between hypodermis and primary cortex. Some of the parenchyma cells in the peripheral layers of cortex undergo differentiation and become meristematic. These cells now represent the core cambium or phloem. The core cambium starts mitotic activity on both the sides, just as cambial ring in the steel. The mitotic activity on the inner surface of the core cambium results in the formation of cells which undergo differentiation into a living tissue called secondary cortex or phloderm just above the primary cortex. The mitotic activity on the inner surface results in the formation of cells which undergo differentiation into a dead tissue called cork or phloem just below the epidermis. The cork covers and masks the hypodermis. The tissue resulting from secondary growth in the cortex, the core cambium and the secondary cortex together represent a region called periderm. The periderm along with the primary cortex represents the bark. In several dicot plants, the bark peels off regularly. Due to the formation of periderm, the epidermis is subjected to pressure, and as a result it breaks at several places to form openings called lenticels. The lenticels, also known as aerating pores, enclose a group of living cells called complementary cells. Through these cells, exchange of respiratory gases and to some extent transpiration takes place. The secondary growth in the cortex results in the formation of periderm. Due to the addition of this region, there is an increase in the girth of cortex. Now, structure of secondary xylem. Secondary xylem is made up of an axial system of longitudinally oriented cells derived from the fusiform initials of the vascular cambium, and a radial system consisting of rays derived from the ray initials of the vascular cambium. In gymnosperms, the axial system consists of tracheids and axial parenchyma, and in many conifers, resin ducts also develop as part of both the axial and radial systems. In angiosperms, the axial system consists of tracheids, fibers, vessel members, and axial parenchyma. Secretory ducts as well as secretory cavities also occur in some angiosperms and contain in different species a variety of substances. Now, secondary xylem of gymnosperms. Gymnosperm wood, which consists primarily of tracheids and rays, is less complex than the wood of angiosperms, which is characterized by greater diversity of cell types and cell sizes. The conifer tracheid has dual functions, providing both support and conduction, and its structure is an adaptation to those functions. It may be very long, reaching 8 mm in some members of Araucariaceae, and on an average is longer than the average length of tracheids in secondary wood of angiosperms. The tracheids of conifers have tapered to relatively blunt ends and overlap those of other tracheids and are characterized by large, usually circular, bordered pits that are very abundant on the overlapping ends. In addition to tracheids, axial wood parenchyma and the epithelial cells of the longitudinally oriented resin ducts are also derived from the fusiform initials in conifers. The vascular rays of conifers, relatively low and uniseriate to biseriate in most species, are commonly composed of two types of cells, ray parenchyma cells and ray tracheids. In species containing resin ducts, the rays also contain epithelial cells lining the ducts, which are large intercellular spaces. Ray parenchyma cells are radially elongated, that is, their radial dimension is greater than their height. Ray tracheids are non-living ray cells that occur in either marginal or marginal and interspersed rows. They are often more radially elongate, and their secondary walls, which contain bordered pits, thicker than those of ray parenchyma cells. Resin ducts are a distinctive feature in the secondary xylem of many conifers. They consist of long channels enclosed by epithelial cells which secrete resin into them. Now, secondary xylem of dicotyledons. The secondary xylem of dicots differs from that of gymnosperms primarily in the greater complexity of tissue and greater structural diversity among different taxa. The most significant difference is the functional separation in dicotyledons of cells providing mechanical support and those which provide transport of water and minerals. Whereas in gymnosperms, both of these functions are served by tracheids. In dicotyledons, support is provided largely by fibers and longitudinal transport by vessel members, except in most primitive angiosperms, for example, members of Winteraceae of Magnolidae, which lack vessel members. The secondary xylem of these primitive taxa closely resembles that of gymnosperms, the axial system being composed primarily of tracheids. Another difference between some gymnosperms and angiosperms is the nature of the pit membranes in the pits of tracheary elements. Whereas in conifers, the pit membranes have a central thickened torus and a surrounding highly porous margo of intertwined cellulosic strands. The pit membrane of tracheary elements in angiosperms is a much simpler sheet of primary wall of uniform structure. The following types of tracheary cells characterize the axial system. Number 1, tracheids. In vesselless angiosperms of the Winteraceae, the tracheids are very long, measuring up to 4.5 millimeter, relatively thin-walled, and bear circular bordered or scalariform bordered pits. They have relatively blunt ends that overlap others in tissues. Now next, fiber tracheids. These cells are of frequent occurrence in primitive dicotyledons, the secondary xylem of which is also characterized by long cylinder vessel members. As their name suggests, they are intermediate in structure between typical fibers and tracheids, although they are usually longer than tracheids in the same wood. Now next, libriform fibers. As the most highly specialized wood fibers, libriform fibers are the most abundant cell type in many woods and are the major elements of structural support. They are characterized by great length, relatively small transverse diameters, thick secondary walls and sharply tapered ends. And next is vessel members. Vessel members, characteristic of all angiosperms except the most primitive taxa, function primarily in the transport of water and minerals. Unlike tracheids and fibers, vessel members have end walls and occur in superposed columns of cells comprising vessels. During development, the end walls between vessel members become perforated. Consequently, vessels are elongate tubes well adapted for the movement of water and solutes in the plant. Now, wood parenchyma. In the secondary xylem, the parenchyma cells are commonly present in two forms, axial parenchyma and ray parenchyma. The axial parenchyma cells are derived from elongated fusiform initials of vascular cambium, and subsequently their long axes are oriented vertically in stem or root. If derivative of such cambial cells differentiates into parenchyma cell without transverse or oblique division, a fusiform parenchyma cell results. The ray and axial parenchyma cells of secondary xylem typically have lignified secondary walls. Now, functions of secondary xylem. The main function of secondary xylem is upward conduction of water and minerals. According to Dixon and Joly's theory, water loss from leaves through transpiration results in the development of tension in the water column in the tracheids and vessels, that is transmitted throughout the column to the roots. As transpiration continues, water is pulled from the soil into the roots and upward through the system. Thus, a water is being lost from the plant. It is constantly being replaced. It is the most important supporting tissue in arborescent dicotyledons and most gymnosperms. Secondary xylem is a complex tissue that consists not only of non-living supporting and conducting cells but also of important living components, rays and axial wood parenchyma, which with those in the secondary phloem comprise a three-dimensional symplastic pathway through which photosynthate and other essential molecular substances are transported throughout the secondary tissues of the plant. Additional increments of this tissue are added during each growing season, usually annually. But in older regions of most woody species, only the outer increments are functional in transport, although the number of increments that remain functional varies greatly among different species. Older increments gradually become plugged by the deposition in them of waste metabolites such as resins, tannins, and in some species by the formation of tyloses. The inner non-functional secondary xylem is called heartwood, the outer functional secondary xylem sapwood. In this presentation, we have learned about formation and structure of secondary xylem. We have also discussed about activity of stellar, extrastellar cambium and growth rings. Thank you. God bless you.